repeat or revise. So first tell me your doubts, the doubts you have. Am I audible to all of you? Muhiddin, Nahara, Mazia?
Yes, sir.
Okay, fine. So please let
Sir,
me
I
know.
have a question, sir. Like
Yes,
it's
sure.
not a doubt, sir.
Yeah.
Sir, which books do you prefer, sir? Like for NEET biology?
For
Like,
NEET,
yeah.
actually, for NEET examination, first I would like to say you if you have access of a book called a Fingertip. So first go for the Fingertip, MTG Fingertip. The publisher is MTG. So first go for the MTG Fingertip. When you complete the MTG Fingertip, just go through with the NCRT, and next the second thing, the previous year question paper, which should be not less than 15 years back, right? So if you'll do this one and NCRT, whatever I have told you, that notes, if you have NCRT, the notes which I have taught you, and if you did the MTG Fingertip, it's not that much thick book, it's this much thick book. So if you complete this, that was the syllabus what I have recommended you, then you will be able to do more than 85 question. You'll get more than 85 question, 36, 37. That is the only thing which you have, right? So
Yes,
try
sir.
to get that book. MTG Fingertip, that is the name of the book, right? Fine.
Okay, sir.
So tell me what I need to revise. What do you want me to revise? Mazia, Nara. Tell me, guys, the chapters which you want to revise or the doubts which you have. No doubt? Mazia, tell me. You can message me even. Nara, am I audible to you? Yeah, cell cycle. Yes, yes, Mazia. That's why I'm late. Actually, yesterday you told me that you'll not come today. You will not join today, and we have to do the cell cycle and cell in the next Saturday. So I thought that the class is on next Saturday. Fine, no issue. So should we start with the cell cycle? So Mohiddin, what's about you? That are the exams are over, or still you're waiting for the paper?
So they are going on, sir.
They're going on, right?
Yeah.
So what about the biology exam? Is it
So it
over?
was today, sir. Yeah, it was
Okay,
today.
it was today. So how was that? Tell me.
It was nice, sir. It was nice.
You attempted all the questions?
Yes, sir.
Fine, fine, great, great. What's about you, Nara? What's about you? Your biology exam? Am I audible to you, Nara? Fine. So Mazia have exam, and she wants me to repeat cell cycle and cell division. So let's start with that chapter on cell cycle and cell division. Yes. So let's start with the cell cycle. This is the cell cycle. So what is the cell cycle actually? So cell cycle is complete division process of the cell cycle. How does the cell divide, then grow, and then they become mature, and then they differentiate, right? So how does that cycle goes on? That is called cell cycle. So let me explain you from this PPT first. So try to understand. So whenever formation of a new cell takes place, try to understand from this diagram. Whenever the formation of new cell takes place, so this is called actually daughter cell. So new cell is called daughter cell. This is a diploid daughter cell. What happened? Now cell starts from a young cell or from the daughter cell. It start its maturity phase. So it's start growing and it become mature. So the time phase between the young state to a mature state, or from a daughter cell to become a mother cell, right? The gap between these two stages is called interphase. And suppose that if a lifespan or a cycle of cell, like division cycle and growth cell, if both of the cycle they takes place 100% time, then 90% of the time is spent in the interphase, that is called growth phase. During this phase the cell grow. So further we have divided this time period, this phase, into three stages: G1, S and G2. In G1 phase cell organelle get doubled. In S phase, DNA synthesis means DNA formation takes place. Protein synthesis means protein formation takes place. And in G2 phase, the protoplasm synthesis takes place, right? After the interphase, when a cell become a mother cell or fully grown cell, what happened? The cell have three choices. The first choice is differentiation. That is, the cell will take a particular function and it will stop dividing. For sake of example, a new cell become a cardiac cell. It start functioning with the heart and it stop dividing. Then, so this is the first option with the cell. Then after the maturity, the second option with the cell is that it pause at that point. It goes for the rest, for the sleeping period, right? And it arrested and work as a reserve cell. Whenever whenever there will be requirement, then it will again start dividing or again enter into division phase. Third thing is that the cell become mature and it will again divide. So this is called G1 phase. When cell get differentiated, we call it G0 phase, right? Or when it become pause or arrested, this is also called G0 phase. But the G1 phase, if the cell is again going to divide, right? So in division, it will take only 10% time, right? So it will enter into Z1 phase. Now it will divide. So there are two kinds of the division. A mature cell may divide by two methods, one mitosis and one meiosis. In mitosis, from one mother cell, one daughter cell formation takes place. From one mother cell, two daughter cell formation takes place. In meiosis, the mother cell have diploid number of chromosome, means 2n number of chromosome, and the daughter cell also have 2n number of chromosome. So we can say in the mitosis, a mother cell, a diploid mother cell divide into two diploid daughter cell. So what is same over here? The number of the chromosome is same in mother cell and daughter cell. So because number of chromosome are equal in mother cell and daughter cell, that's why this is called equational division, because the number of the chromosome are equal in mother cell and daughter cell. The second type of division is called meiosis. A mother cell, a diploid mother cell divide into four haploid daughter cell. So here the number of daughter cell is just double, not 2, 4. In meiosis process, a mother cell divide into four daughter cell, but the number of the chromosome are diploid in mother cell, but they are haploid in daughter cell. Means the number of the chromosome are half. So this is called reductional division, right? The number of chromosome get half, right? And this division state is called M phase, right? And it takes place 10% time, right? So this is this complete cycle 90% time which is taken by the interphase, and the division phase which is called M phase, right? This complete cycle is called what? Cell cycle, right? Is it clear or not? Tell me, Mazia. Is that clear with you?
Yes.
Great, great. So if you want, you can take the screenshot as well, because you won't find this kind of the cycle in any book, right? I have just explained you, so this is one of the best way to explain. If you want, you can take this screenshot. Right. So now we'll study one by one. First, we'll study the interface. What actually takes place in interface? How does the division takes place in interface? Right. So I'll tell you one by one. Right. What are the processes which take place? So first, the next topic is that the stages of cell cycle. Now there are certain stages of cell cycle. Look, this is the stage of cell cycle, right? So the interface, the time period between two division phase. So first phase is division. One more thing I want to tell you. Cell division takes place generally in two stages. First, karyokinesis, when the nucleus of a cell divide, and karyokinesis takes place in two manner: mitosis, when the number of chromosomes remain equal, and meiosis, when number of the chromosomes get half. Then, when nucleus divide, after that cytoplasm divide, and that is called cytokinesis. That is the protoplasm, right? And the Fleming was a person who given the term mitosis, and Farber and Moon were the person who given the term meiosis, right? So this was meiosis and mitosis. And the next thing that here, after mitosis, when cytokinesis takes place, the G1 phase is called gap 1 phase, right? Gap 1 phase. The cellular content, excluding the chromosome, are duplicated, right? So DNA formation do not take place, DNA synthesis do not take place. They get duplicated, so they become just double. After G1 phase, there is a checkpoint. What is a checkpoint? Checkpoint is just inspection, right? When you enter in an airport, there is a checkpoint that check everything, that are you eligible to go ahead or not. If you are eligible, then you will go. If you are not eligible, you won't go, right? So that, like the same way, there is a guard, that is a protein, and the name of the protein is CDK, cyclin dependent kinase. That is a checkpoint. What it will do, it will check each and everything, that is the cell ready to go ahead or not, right? If the cell is ready to go ahead, then what will happen? Then it will be allowed to go ahead. Now what will happen? If it's okay, then it will go ahead. So this is the first checkpoint, and this checkpoint is called G1S checkpoint, because the checkpoint is between G1 and S. Now cell will enter into another stage called SS stage. S means synthesis. G1 means gap 1 stage, right? Gap 1 phase. S means synthesis phase. So what will happen in synthesis phase? In synthesis phase, the synthesis of protein takes place, synthesis of DNA takes place. So both of the synthesis takes place. Then in G2 phase, what will happen in G2 phase? In G2 phase, the DNA replication stops, but the RNA and protein synthesis keep on going, right? After the G2 phase, crossing the G2 phase, there is again another checkpoint. Again, there is a guard, and what is the name of the guard? This is the same guard, CDK, cyclin dependent kinase. This CDK, this is the CDK. This CDK protein check that is that cell is prepared to go for division or not. If it is prepared to go for the division, then it will allow it, and it will further go for the division. If it is not allowed for a further growth or further division process, then it will be stopped over here. This is called G2m checkpoint. Why? Because this is the checkpoint between the G2 phase and M phase. Now, after the G2 phase, if it is okay, then it will go for division, and it will take 10% time to divide. Either it will divide by the mitosis or meiosis. First, karyokinesis takes place, then cytokinesis takes place, and then will divide. So this is the sub stages of interphase G1, S, and G2. Right. Come to the next part. So the next part is look. So the first phase is G1. Now, what happened to the G1? Try to understand what happened with the G1. So in G1 part, in G1 phase, this is the longest phase, right? This is the longest phase, means it takes too much time, right? So this is the longest phase of interphase, right? RNA and protein synthesis takes place during the G1 phase. Nucleotide, amino acid, histone protein which remains associated with the, which is like chromosome are made up of two things, DNA and histone protein. So this is a basic kind of the protein. So nucleotide formation takes place, amino acid formation takes place, histone protein formation takes place, and ATP formation takes place, which are called energy currency, right? In G1 phase, the duration is variable, right? It depends on cell to cell. G1 phase may be longer or shorter, depend on the cell to cell, right? Next, that is the S phase. What is the S phase? In S phase, now when the cell enter into S phase, this is called synthesis phase. Here the chromosome replicate. So this is the chromosome. So number of the chromosome do not increase, but their genetic content, means DNA content becomes double, right? Look, this is the one chromosome. Again, this is also one chromosome. So number is one, but here the extra arm formation takes place. So the DNA content get double, but the number remains the same. Here the chromosome replicate and DNA become double. This is called invisible phase. Why? Because here the number of chromosome do not increase. Inside the chromosome, the genetic material become double, right? That's why, because this phase is not visible with the microscope, that is called invisible phase. Why? Because there is no visible change which you can see with the help of your microscope or electron microscope. That is not visible. So that's why I call invisible phase. Here, the kinetochore synthesis takes place. So kinetochore, there is a type of protein which is found on the centromere. This is called kinetochore. Again, the another phase is called G2 phase. Here, the synthesis of DNA stops and RNA and protein synthesis continue. So this is called G2 phase, right? So that's why that's how there are three kind of the phases which are over here, right? So tell me, is it clear or not? Clear, Mazia? Keep on messaging me. You can
Yes.
message. Now there is one more concept. So first one is, now come to the M phase. We have studied interphase. Now we are here to understand the M phase. Mitosis. So mitosis is like the cell is in interphase. The first one. This is in interphase. So here, this is a centrosome, and centrosome start in centrosome. There are two small T-shaped structure. These are called centrioles, right? So this structure is in protoplasm. This is called centrosome, and inside centrosome there is a structure called centriole, right? This is the nucleus. Okay, this is the nucleus, and inside the nucleus there are chromosomes, and this is a small structure which is called nucleolus, nuclear membrane, and another thing, the outer structure, that is called plasma. What happened? This is the centrosome. Inside the centrosome there are two centriole T-shaped structure. Now the cell enter into prophase, right? So in prophase, what happened? The early mitotic division, whatever happened over here, uh, the this centriole start dividing and they start moving toward the opposite pole. So these centriole reaches to centrosome, divide, and centriole reaches to opposite pole, right? Nuclear envelope start disintegrating, right? It disintegrates, and two sister chromatid or single chromosome, they get condensed and they become chromatid, right? Here, what happened in pro-metaphase? This is the initial stage of metaphase. In pro-metaphase, the nuclear envelope disintegrate, right? It divide, right? It get disintegrated, and again, the formation of spindle fiber takes place from where? From the centromere, right? Formation of spindle fiber takes place. These spindle fiber attach as the kinetochore, right? Where the kinetochores look. Here I have tried to show you the actual structure of the chromosome at this stage. Here the genetic material is already double. The central part is called centromere, and both of the side of the centromere there is a disc type of the protein that is called kinetochore. There is a kinetochore. A spindle fiber attaches with the kinetochore, right? What happened? The spindle fiber attaches with kinetochore. Again, it enter into metaphase. When enter into metaphase, these monovalent, or we can say these chromosomes, they arrange in the equatorial plate. This is called metaphasic plate or equatorial plate. So when you see the equatorial plate, this is called metaphasic plate. Basically, we see the chromosome with the help of microscope. To study the chromosome, we study the metaphasic chromosome only, right? So the picture which you see in your book, your notebook or NCRT, that is the metaphasic chromosome only, because the chromosomes are clearly visible at this stage, right? After the metaphasic place formation, what happened? This spindle fiber I want to mention over here. The spindle fiber are basically made up of a type of the protein, and the name of the protein is tubulin protein, right? This is the tubulin protein. What happened? The spindle fiber start pulling the chromosomes to the opposite pole, right? They pull the chromosome toward the opposite pole. These daughter chromosome reaches to the opposite pole, and this is the anaphase. After the anaphase, both of the chromosome reaches at their end, and the nuclear division or karyokinesis is complete, right? In the karyokinesis, again, this stage, like telophase stage, is the reverse of the prophase. What happened? Again, spindle fiber formation, spindle fiber disappear. Centrosome, this centriole reaches at their pole, right? And again, they divide and make a centrosome, and nuclear envelope again reappear, and two nucleus get divided. Now it comes to the part of cytokinesis. Cytokinesis, till this point, the division of the plant cell and division in the animal cell, like mitosis in both of the plant and animal cell, is similar. But means the karyokinesis in plant and animal is similar, but there is a difference in cytokinesis. If this is the animal cell, please mind it, if this is an animal cell, then what will happen? Formation of a furrow takes place from outside to inside. So this is a furrow formation by the centripetal manner. So in animal cell, formation of furrow takes place. These furrow meet at the end, and after that, the cell divide over here into two parts. The another division in animal cell, also in plant cell. So what happened in plant cell? If suppose that this is a plant cell. In plant cell, at the center, a material deposit over here that is called phragmoplast. So phragmoplast deposit, and it makes cell plate. Formation of cell plate frame takes place from center to outside, means this is centrifugal. So in animal cell, the protoplasm division takes place in centripetal manner, and initially it start as a furrow formation. In plant cell, the cell plate formation takes place, which is a centrifugal manner, and it takes place due to formation of phragmoplast. So that's how after that, after some time, this cell plate become complete and it divides cell into two parts. So this is the division in the plant cell. That's how the division takes place in plant cell and animal cell. So this is all about the mitosis. And please let me know one more thing: a spindle fiber are made up of 4 to 20 microtubules, right? So tell me, is that clear? And if you have, look into your NCRT. If you have any doubt, please ask. Mitosis. It's clear. Okay, fine. Okay, Nara, your exams will start from 17. So yes, Nara, after this cell cycle, if you want, like if you have doubt in any chapter, then you can ask me right now. Because 17, you have one more class. So you can take one more class, right? So in this class, ask the doubt, and even next class, join the class and like ask your doubt, clear your doubt. It will help you a lot. So now it comes to the meiosis. You know, Nara, if they will ask you a long question from this chapter, definitely they'll ask you meiosis, right? Generally, teacher have tendency to ask meiosis as a long answer. They hardly ask for the mitosis. But I cannot deny the fact if the question paper will be easy, they can ask you the mitosis as well, right? So let's start with the meiosis. So look, the first meiosis takes place in two parts: meiosis first and meiosis second. Meiosis second is exactly like the mitosis, whatever we have studied, but meiosis first is a bit different from the mitosis. Look again. So come to the meiosis first. This is called reductional division. Why? Because in meiosis first, the number of chromosomes become half. That's why we call it reductional division. So this is an interphase nucleus. So in interphase nucleus, this structure is called centrosome. In prophase first, what happened? This nuclear envelope disintegrate or divide, right? It dissolves. Again, the two chromosomes come into pair with each other, and we call this structure bivalent. So formation of bivalent takes place. So first thing, what? Nuclear envelope dissolve, disintegrate, it got broken down. The second thing, formation of bivalent takes place. Again, after that, the centrosome divide and centriole start moving toward the opposite pole, right? Now come to the metaphase. In metaphase, the formation of metaphasic plate takes place, means these bivalent get arranged in the equatorial plane, right? And these both of the chromosome pulled in opposite direction, and the number of chromosome get half. That's why this is called reductional division, right? So from one diploid cell, formation of two haploid daughter cell takes place here, right? So in anaphase, both move in opposite pole, and after anaphase, there is a telophase. Telophase may or may not be there, but most of the time it remains. Again, the cytokinesis takes place. Now a cell divide into two haploid cells. Now in meiosis second is called equational division, because now they are the mother cell. So there are two haploid mother cells, and they will form four haploid mother cells. So now four haploid daughter cells. So the number of the chromosome remains equal. That's why this is called equational division. So this is the telophase stage. After the telophase, they directly enter into prophase second. In prophase second, again, what happened? The nuclear envelope disintegrate. Again, this centrosome divide and centrole moves in opposite pole. In metaphase second, they get arranged in equatorial plate, right? In anaphase, they are pulled opposite direction, and in telophase, again they divide into four cells. That's how the meiosis takes place. But the meiosis first is called reductional division because number of the chromosome become half, and meiosis second is called equational division. Equational division because the number of the chromosome remains equal. Is that clear? Tell me, guys. Is that clear or not?
Yes.
So this was meiosis. Now, especially, I want to tell you one more thing. This meiosis prophase is further divided into subphase because this is the longest phase of this meiosis process, right? So now I am going to tell you all the incidences which takes place in meiosis first. Further, we have divided meiosis prophase into further 5 parts. So what are those parts? In meiosis first, right, prophase first, it is divided into leptotene, zygotene, pachytene, diplotene, and dikinase. What happened? In leptotene, nuclear membrane start disintegrating, and the chromosome get condensed and they become more thick, right? In zygotene, formation of bivalent takes place. And what happened during the bivalent? The sum of the part of these non-sister chromatid, they get attached, and this attachment, suppose that there are two chromosomes, so they get attached with each other, so they make this attachment is called synapses. So formation of synapse takes place, right? And this structure is called synaptonemal complex. So the formation of synaptonemal complex takes place between the two chromosome or two non-sister chromatid. Now what happened in packaging, these both of the chromosome arm, they exchange their part each with each other, and a crisscross structure formation takes place. This structure is called, the changing part is called crossing over. So crossing over takes place, keep it in mind, in packet species crossing over takes place where the chromosome exchange their part, right? After the crossing over, they start separating, and during the separation, this Caesar like structure formation takes place, crisscross structure formation takes place. This is called synaptonemal complex. In synaptonemal complex, there is a chiasmata, right? This crisscross structure is called chiasmata. So in diploid stage, formation of chiasmata takes place, this crisscross structure, right? And now the synaptonemal complex, it dissociates, so the chromosome starts separating, right? In dikinesis, nuclear membrane disintegrated finally, and the prophase get end. That's these are the sub phases of meiosis prophase first. So is it clear? Crossing over, synaptonemal complex, chiasmata, all those terms, are they clear to you, Mazia and all?
Yes.
So this was all about this chapter. So Mazia, look into your NCRT and let me know if you have any doubt in your NCRT. Just have a glance on your NCRT. This is very important concept for your medical examination as well, parallel. And Nara, tell me if you have doubt in any of the topic. Okay, Bazia, no more doubt. What's about you, Nara? Tell me.
No, sir.
No doubt. So all doubt clear. Tell me anything else you want me to repeat. You want to study anything else? Even Mazia, look into your NCRT. If you have any doubt in your NCRT, ask me. And same with you, Nara. Then, because this is the right time to ask, you have ample of time. And again, what I am saying is, again, you study and revise the concept. And in case if you have any doubt, then you can, like even you can arrange the class before Saturday. Whenever you want, you can arrange the class. Right? You can coordinate with Niskarji. You can take time from me, and we can arrange the class. And again, there will be a revision class on Saturday, next Saturday as well. So that is the regular. But if before the Saturday, if you need my class, if you need any help, then you can contact to Niskarji. Will help you out. No issue. Right? So just have a glance with the NCRT again. both of you, Mazia and Nahara. And if you have doubt, then let me know. Even if you don't have doubt, then let me know. Yeah, sure, 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 fine. No issue, Mazia. And again, go with your syllabus. And if you have any doubt, meet me on next Saturday, right? What about you, Nara? Message me, please. Yeah, welcome, welcome, Mazia. Nara, ask me if you have any doubt, then we'll leave. If you don't have any doubt, then I will wind up the class. Fine, Nara. So again, it's the same thing for you. You have 5 days time. So if you have any doubt, then if you don't have any doubt right now, so do one thing: revise the complete syllabus and note down the doubt. Right? Definitely, yeah. Note down all the doubts in one page, right? And then next Saturday, ask me. Fine. So till the time, bye bye. Take care. All the best. Yeah, definitely. Ask me.